working with type 2S assembly, and we picked Juniper GFP. Is it a good choice? So I personally haven't worked with Juniper GFP before. So I don't know, if it has any particular, oddnesses to it. If you're working with only one fluorescent protein, then typically it's much easier than if you're trying to work with multiple. If you're working with just one fluorescent protein, then you don't have to worry about overlaps and things like that. So the thing to check is basically check its maturation time. If its maturation time is in hours, you might want to worry. If its maturation time is a few minutes, then it's easy to interpret. And to check its brightness. And in terms of brightness, if its brightness is on a fairly similar level to other GFPs, then you're probably fine. If it's very dim, then you might have some trouble with it. But check out those things. FP-Base probably has it. And if you have more questions, feel free to email the measurement committee after. All right. See another question that says, different colors are often used to show reaction when induced. For example, green. We're going to clone a regulator gene in selenomycin. For example, what would you use in such a case? I'm not sure if I understand this one. So if you're saying choosing which color would we want to choose in order to show a particular reaction, one of the nice things about fluorescence proteins is that typically you have a lot of flexibility in them, because in a lot of cases you can fairly easily substitute one for another. So which one to use really depends most on what system you're going to be measuring with. I typically, if you're going to measure only one protein, I typically recommend starting with a green, something. In particular, I recommend measuring something that you can calibrate using fluorescence. So like a GFP or a YFP, something very similar to that green-yellow fluorescence color. The reason I recommend that is because fluorescence is the cheapest and easiest calibrant to work with. So if you have to pick just one calibrant to work with, that's a nice one to go with, because it's way cheaper and has no solubility challenges or anything like that. So there's often a free choice. If there's a free choice, I choose green as my first starting point. All right. Seeing no more questions at the moment, let's move forward to the second section. Now that we've talked a little bit about fluorescence in general, let's talk specifically about how to calibrate a plate reader for fluorescence and OD measurements. So the thing that we're going to recommend to you here is the iGEM plate reader calibration protocol. And we call it the iGEM protocol because it was developed by iGEM teams over the course of a couple of years of interlaboratory studies. The nice thing about this protocol is that it doesn't use anything tricky. It uses cheap, stable materials. For calibrating GFP, we've validated that you can do it with fluorescence. Fluorescence is incredibly cheap for a laboratory reagent. I think it cost us something like less than $100 to supply all of iGEM with kits of fluorescence. So you can buy one sample and it'll last you forever. Red fluorescence protein, Texas red. There's a specific one. I've got these sigma numbers will lead you to the right version. There's a bunch of versions of Texas red, and some of them are not soluble. So you want to get the one that is. So those are what you want to use for your fluorescence. We have a potential recommendation for blue, but we haven't validated yet. For calibrating OD into an equivalent number of cells, you basically want to use silica beads that are the same size of the cells. The ones recommended here, this NanoSim 950 nanometer beads, those are ones that are good for calibrating E. coli or other similarly sized bacteria. If you want to use some, if you're working with a different type of cell, like say yeast or mammalian cells or plant cells, you want to use bigger beads that are similar in diameter to your cells. And this is of course, the cells are not really round, but this is what we call a spherical cow approximation. You know, assume that the cow is a sphere. Assume that the E. coli is round. How big would it be? This protocol, when we run it, produces a molecules of equivalent fluorescein per cell or molecules of equivalent Texas red per cell measurement. That one of the nice things about it is that it is directly comparable with flow cytometry. Down on the bottom left, you can see results from the iGEM 2018 interlab study comparing flow cytometry measurements and plate reader measurements in the methyl per cell measures. And the thing that I want you to notice there is that these measurements are the same. It's also a highly replicable and debuggable measurement. The iGEM teams previously got to within about 1.8-fold, which is a very nice number. And like I said, you know, hundreds of iGEM teams have used these. So this is a very accessible protocol. The protocol can be found up on the measurement protocols page. And the idea is basically that you do a serial dilution of the particles and each dye that you're working with. And then we have some Excel sheets that you can basically just paste your plate reader measurements into in order to calibrate. You paste the calibration measurement that your serial dilutions in order to get the conversion factors. And you paste your experimental data in on another sheet in order to take your arbitrary unit measurements and convert them into MEFL per cell estimates. One thing though, this is true not just for this protocol, but for any protocol, some instruments will try to adjust themselves for you, and you cannot compare two measurements that are made when the instrument has different settings. So you have to make sure that running your calibration and running your experiment, your instrument has the same settings. And that means that you have to make sure your instrument is not trying to auto-calibrate for you, because if it's trying to auto-calibrate, it will change the settings on you. While I personally would love if people reran calibration every single time, that's not really practical. I recommend weekly, but doing it at least monthly can ensure that you check to make sure that nothing has changed on your instrument. So the specific idea here, you start with a concentration of stock, you know, in the OD at a certain number of particles per milliliter for the fluorescence at a certain micromolar concentration. These numbers are relatively arbitrary starting points. They're starting points that we chose essentially because we found these to be useful starting points. But if you have some need to adjust them, there's nothing magic about these starting points. These are just what the sheets are set up for. And just transfer your stock into these wells that you've filled part way with blank material, and then dilute by half each time by transferring from well to well to well. Do this in replicate. We recommend quadruplicate. And at the very end, there's one that you just never put any material into. So you should get a nice even dilution like you see up in the upper right. And this should be adaptable to other cell types and other fluorescents as well, just by changing the calibrant that you're using, as noted before. So what's it look like when you get a nice calibration? Here's a couple of examples. This is real data from the Interlab in 2018. On the left, that's a particle example. This, notice, so these are all on log-log scales, and a good example will have a nice long region somewhere that is on a diagonal. That nice on the left, it's a nice long diagonal, which then saturates as it comes to the detection limit of the instrument, and that's okay. What that saturation is telling us is where the instrument can't detect anymore. So this is telling us that each OD unit coming off of the instrument is equivalent to 3.26 times 10 to the 8th particles, and that this conversion is valid when those AB 600 arbitrary units of OD coming off the instrument are more than about 0.043. So that's the point where we enter into the range that we can't determine the difference between 0 and our measurements anymore. Over on the right, the fluorescence curve is linear the whole way. You'll sometimes also see something that saturates at the top when you're hitting the maximum intensity that the instrument can see, and it starts overflowing. It's actually fairly typical for the range of fluorescence that can be measured to be larger than the range of OD that can be measured. That's okay. So this is a fairly typical example here. Here's some examples of what you might see if it's not so good. So if what you're seeing is not linear in the log scale, except where it saturates low or saturates high, you've got a problem. On the left is an example. So see that sort of wiggly? That looks like somebody didn't have a very good day pipetting. That probably just needs to be redone. Over on the right, this one's a little bit weirder. It looks almost right, except that there's this spot where it goes boom, way up. And my guess is that that is actually a problem in data entry, that somebody may have slipped a decimal point there. So if you see something funny, here's some examples of how it can go wrong and what might be causing it. Another thing that we see very often as a way that calibration can go wrong is that you end up with a narrow range. Here we're seeing that from the very highest density to the point where it enters the instrument noise is only about a 4-fold difference. So technically you could work with this, but this is telling you that the instrument settings are probably wrong. The instrument settings need to be changed, or maybe the media needs to be changed some way or other. We're not being able to see small amounts of cells. And so something needs to be adjusted in this protocol. Typically, you'd want to see at least a 20-fold range from high to low. And you can have yet other values. Here's an example where somebody was getting negative values out of their OD. The readings that you get for these should always be positive. There is never, you can never make water more transparent by adding particles to it. And you can never make water unfluoresce or negative fluoresce. If you get negative numbers, that probably means that there is a problem with your instrument, typically something that may need maintenance or reconfiguration, or else somebody's done something very strange. With another way you can get negative numbers is if it's a negative difference between your actual numbers and your blank, and that might mean that you got your blank contaminated somehow. Yeah, negative values are always a bad sign. We have some software available if you are interested that can help with finding these types of debugging. I put a link here to the Excel process validator. This is something that can be run in MATLAB or Octave against your Excel sheet. And it basically looks at those Excel sheet templates that we provided and we'll check for a bunch of the common problems that can be there. And this is an example of the output that it's giving for a good run, saying that it found sufficient dynamic range, nice long dilution slopes, significant cell counts, etc. So I want to end on this part by talking a little bit about how to interpret what these units actually mean. Let's start by talking about this equivalent particles unit that we get by calibrating OD. The calibrated units that you get are pretty close to cell counts, but they're not the same as cell counts. Again, first of all, our E. coli are not actually round. The silica spheres are an approximation, but there are also some things that can happen. I was about to say go wrong, but it's not necessarily go wrong. There are things that can happen in your sample that can affect the apparent density of cells without changing the actual cells. First is if there's a bunch of debris in there, say from dead cells, you may have a bunch of junk that's making your sample cloudy and therefore increasing its OD that's not cells. A second thing, the scattering model for OD assumes that your cells are dispersed throughout the sample. If they're packed into a layer, a thin, tightly packed layer can block light much more effectively than the same amount of material spread out. You can see this same principle just by looking at a piece of fabric. So if your cells are forming a biofilm, or if you're working with an adherent cell type, that means that your OD measurements can't be interpreted directly in terms of number of cells either. And a third thing is that we're estimating based on particles that are as opaque as a normal cell that's not filled with very opaque stuff. If you express something in your cells that is a very opaque material, the LAC-Z reporter is a nice example of this, that basically fills your cells with a dark pigment. That will affect their opacity, and thus you may estimate that there are more cells than there actually are. Still, if you're working with cells in liquid culture that are not expressing anything terribly opaque and are in a relatively clean medium, this is a pretty good estimate of number of cells. Similarly, the fluorescence, we get a molecules of equivalent something, whatever our calibrant is, molecules of equivalent fluorescein, molecules of equivalent Texas red, which are close, but not the same as molecule counts. This is because fluorescein is not GFP. They have different excitations, different emissions, and those differences matter, even though they're not huge. And the farther you get from the ideal measurement ranges, the more that those differences matter. There are also issues, like we said, if you have a very slow folding molecule and you measure it before it's had a chance to fold, that will make it look apparently lower fluorescence than the actual count of molecules we have. Similarly, if your colonies are not well oxygenated, this will cause there to be less apparent fluorescence than actual molecules, because the molecules need oxygen to mature properly. Still, under sort of typical conditions, with good oxygen, good shaking, and normal measurement with a fast folding thing, you've got a pretty good close approximation. So let me summarize, then we'll pause to take a look at questions again. Calibrating plate readers can be simple, cheap, and reliable. Make sure that the settings on your instrument are the same. Make sure you turn off those automated calibrations and recalibrate at least monthly, and you'll make me very happy if you calibrate once a week. Doing calibration can detect problems with your instrument, your configuration, all sorts of other things that can go wrong in your lab. We have some automated validation software that you can use for that if you wish. Finally, the units that you get are close, but not identical to cell count and molecule count. And you should be aware of some of the key types of condition that can cause a large value change. With that, let me pause a moment and look and see if there are any questions that come through. Okay, seeing no questions at the moment, I will move forward.